Hello. Thank you so much for this opportunity to present my work here. My name is Shruti, and my PI is Dr. Anand Jayashekharan, and I'm a third-year PhD student. So I'd like to start off my presentation by showing you all these two images. Each of these belong to two separate patients, patient A on the right, patient A, and patient B. Now, both of these are images of the patient biopsies, which were then sent to the pathologist. And the pathologist looked at them, scored them, and gave the final diagnosis. But then both of them got the same diagnosis, but patient A did not relapse, but patient B did. So it was two patients, one diagnosis, but two vastly different outcomes. And this was the case for over 40% of diffuse large B-cell lymphoma, or DLBCL patients. And diffuse large B-cell lymphoma makes up a large fraction of non-Hodgkin's lymphoma, around 40% of cases. So this is a pretty big problem that we would like to solve. And we kept thinking, what are these slides telling us something extra that we're not able to see? So we came up with the hypothesis that cancer basically has its own real estate going on. As Professor Gobun previously said, we've gotten really good at figuring out how cancers behave as a clump of cells, right? We can sequence them, we know how they behave. We have all the technology, which is becoming cheaper and cheaper by the year, to sequence them as a group of cells. But maybe it's actually such a vast neighborhood, and it's actually very complicatedly arranged in different neighborhoods and sections. You can have some areas where there are dense clusters called cluster lanes of cancer cells, some which don't have cancer cells at all, but just have other cells that are arranged. And maybe it's actually organized in a very intended fashion that and it's cancer's real estate. And just like in real estate, it's location, location, and location. And perhaps this was the missing piece that the slide was telling us that we can now piece together. And that formed the basis of my PhD, to try and see if I can now map tumors like cities. And like my PI and our lab likes to say, historically, we have mapped seas through cartography and charted them and navigated them. We are cancer cartographers. We navigate cancer by creating maps of cancer. And this is another image of a biopsy. And this is how the map would kind of look. It's a little complicated, but each of those green dots represent one cell. And now with an entirely new field of mathematics called spatial statistics, I'm able to work around and see how close these cells are, how far they are, and create neighborhoods from these cells. Broadly, this field is called spatial biology, where different cells in the tumor are essentially like a city map. So I went ahead and I did this for a large cohort of patients. And what I found was, if I have the same cell, but they're on a different street, I have a different outcome. And what does this mean? So I have one case where the cells are extremely clustered together. And in this case, it's a great, good zone. People have better prognosis, people survive longer. But at the same time, if my cancer cells are spread apart, this immediately becomes a high-risk zone, and patients have a poorer prognosis. And not so surprisingly, patient A, who did not relapse, was of the clustered type, and patient B was of the dispersed type. And I've been able to show this consistently across a cohort of 449 patients initially, and now scaled up to over 1,000 patients where the dispersed tend to do worse, clustered tend to do better. So the natural next question was why? So dispersed neighborhoods, no COPS equals no control. And the COPS here are very simply the immune cells. So I have my clustered cells, my dispersed cells. The cluster is filled with immune cells that are able to exert control and some modicum of organization in the clustered tumors, but the dispersed cells barely have any immune cells. Now, the same 3 cells are basically doing the work of 20, and to be honest, they're getting exhausted. So you have no COPS, overworked COPS, things slip through the cracks, and you have worse prognosis. And it's very interesting that we're able to make such immune associations just from a slide based on how cells are located. So I'd like to call this more than meets the slide. So current practice, traditional pathology looks at what cells are, not where they are. But the reimagined future is now we add that missing piece of the puzzle, and we tell people how they're arranged in space. And in the future, hopefully different neighborhoods can mean different treatments. So one step forward in this, we wanted to create a tool that pathologists can now try and use and make it as user-friendly as possible. And with all the data that I've shown you, it sounds like a great problem for machine learning and artificial intelligence to solve. So that's exactly what we did. We created something called the SEDAL calculator, which is like a very simple web app where a pathologist can go and it's drag and drop, and you can input in your percentages that you see on the slide, and the app kind of predicts if the patient is going to relapse or not, and what's the probability of relapse. So now we're testing this out, and hopefully with a lot more real-world data, we can be able to make this a lot more robust and integrated into existing workflows. I would like to thank the entire ADJ lab for this. None of this would have been possible, and especially the Diana Koh Foundation for giving me a chance to reflect on my work and see where it all fits in. Thank you.